Started. I don't know what it is about these things, but I'm nervous and I'm not even presenting. So, good afternoon, everyone. Thanks for coming out, right? Spending your Friday afternoon to attend Matthew's defense, right? So it's my pleasure to introduce him, right? So he's a BME major, also minoring in biochemistry and Russian, which I think again just highlights, right, kind of this diverse, right, set of interests here. So for those of you that haven't met before, I'm Carlo Malaga. I'm an assistant professor in the Biomedical Engineering department, and it's been my pleasure to have worked with Matthew over the past 2 years on this work. Okay? In that relatively short amount of time, he's been able to accomplish quite a bit, which you're going to see just a fraction of, to be honest. I think one of the biggest standout things that I learned working with Matthew over this period of time is just how his intellectual curiosity. That was one of the things that really stood out to me. A perfect example of that is he actually went on a little side quest to make a EEG-controlled drone, completely unrelated to this work. He did it just for fun, right? And so if you're interested in that, that can be a follow-up discussion. Okay? And so I'll just end things here with just saying it's truly been an honor, truly been a privilege to work with Matthew. And assuming he still kind of sticks with this moving forward, I'm very excited for a future colleague. And so with that, I'll let him take it away.
Wow. Thank you so much. Thank you for that introduction, Dr. M. I'm super excited to be presenting my research to you guys. And so just thank you to you all for coming here at 3 p.m. on Friday to see this. It means a lot to me. So without further ado, I'm going to talk about my research, and my talk is entitled The Comparison of Thalamic Atlases in Defining the Motor Subnuclei for Deep Brain Stimulation of Essential Tremor. So in this presentation, I'm going to first begin by giving an introduction into deep brain stimulation and talking about the terminology. Then I'm going to talk about the comparison of thalamic atlases and segmentation techniques that I did. And then I'm going to talk about the impact of this work and kind of a future translational potential of it. So I'll first begin by talking about deep brain stimulation. So essential tremor, or ET, is a neurological disorder characterized by uncontrollable shaking of the upper extremities. This is a common tremor disorder and affects 1% of the population worldwide. So essential tremor is diagnosed clinically. So you go to your neurologist, they have you perform a series of tests, and then they make the diagnosis of essential tremor. So you can see on the slide the spiral drawing task. That is one of the tests that they employ. You can see the healthy control differs substantially from the essential tremor, and you can see those tremors manifest when they draw spirals. And so the origin of essential tremor is mostly unknown. So the treatment options for essential tremor first include medication. That's the first line of defense. So medications such as beta blockers and anti-seizure medications are used. In addition, adaptive devices are used by essential tremor patients. These include weighted items placed on the wrists and also anti-vibration devices that cause tremors to be mitigated. So if both these treatment options don't work and are limited in their effectiveness, surgical interventions are used. So focused ultrasound and deep brain stimulation surgeries are used. And so I focused on deep brain stimulation. So deep brain stimulation, or DBS, is a treatment for essential tremor and other movement disorders. So DBS involves placing small electrodes into the brain and target precise regions of the brain. So if any of you have heard of like a pacemaker for like the heart, this is like a pacemaker but for the brain. So you have your pulse generator located in your chest, a wire that runs under the skin up to the top of the skull, and then a lead or very small thin wire is placed down into the brain. And so at the end of the lead are electrodes. So you can see right here some of the electrodes. And when these electrodes are turned on, small amounts of current are emitted from these electrodes. And when stimulation is turned on in the correct region, these patients that previously had tremors see that those tremors go away. And so while the efficacy of DBS is undisputed and well established, there's a lot of uncertainty regarding the exact mechanism of action. And so here's an example of DBS. So the person in the center, she had the DBS implant, and so her doctor is going to turn off the implant briefly, and you can see her tremors go away, or her tremors begin to start. And then as he turns off the implant, you can see the tremors go away. So this is an example of how you can see in real time the tremors are going away when the implant is turned on. And so the precise area that the lead is implanted varies on the type of condition that the surgeons are trying to treat. So in essential tremor, the surgeons locate the lead in the ventral intermediate nucleus of the thalamus. However, for Parkinson's disease and dystonia, they locate the lead in the subthalamic nucleus, or STN. Also for Parkinson's disease and dystonia, they locate or place the lead in the globus pallidus interna. However, in my data set, I'm focusing on essential tremor. So the relevant target for essential tremor is the ventral intermediate nucleus of the thalamus. So just to give some background about the thalamus is a major relay center in the brain, and it's one of the basal ganglia. And since it's one of the largest of the basal ganglia, it's conventionally broken down into smaller pieces called subnuclei. So the ventral intermediate subnucleus is one of the common subnuclei that are targeted in deep brain stimulation for essential tremor. And so you can see in the image at the bottom of the screen, that's the thalamus, and the different colors are different subnuclei of the thalamus. One of the main ways that you can break down the thalamus, and one of the main categorical ways to separate the subnuclei, is between motor and sensory subnuclei. So the motor subnuclei are involved in movement, so motor pathways. And so those subnuclei are involved in signaling for those pathways. The same thing for sensory. So sensory subnuclei are involved in signaling in sensory pathways. So that involves touch, feel, that sort of thing. And so when the lead is implanted into the brain and the electrode is turned on, the surrounding tissue becomes activated. So all the tissue within that kind of range of activation are directly modulated. However, those neurons that are modulated may interface on neurons that are outside of the field of stimulation. So while you do have local effects, there's also a network level effect of the stimulation. And so while DBS has great outcomes, there's also some side effects that are seen in patients. So the ideal is the tremor reduction outcome. So a patient comes in, the surgery is performed, and then eventually, after the surgery, the implant is turned on and the tremors go away. However, there's also sustained paresthesia side effects. So a sustained paresthesia is a tingling or numbness in the extremity. So if any of you guys have had your hand fall asleep or leg fall asleep and you feel that kind of like tingly numbness sensation, that's what a paresthesia is. And so for these patients that have this side effect, it's sustained. So it's not momentarily, it's for a long period of time. Other side effects include speech difficulties and muscle contractions. And so when the DBS lead is placed in the thalamus and stimulation is turned on, when that volume of activation is in the motor region, as seen in the red region of the thalamus, we would expect to see a tremor reduction outcome. However, if the lead is implanted into the sensory region, so the blue region on the thalamus, we would expect to see a sustained paresthesia outcome, which is a side effect that we don't want to see. And so current clinical MRIs don't have the resolution to differentiate between the subnuclei of the thalamus. So if you look at the 1.5 T or 1.5 Tesla MRI to the right, the thalamus, which is outlined in red, you can see it's homogeneous throughout. You can't really differentiate between the subnuclei of the thalamus. As you increase the resolution and the strength of the MRI to 3 Teslas and 7 Teslas, you start to get to see some differences. I don't know how well it's showing up, but you get to see some differences in subnuclei. However, you can't differentiate between all of the subnuclei just from imaging alone. And the 7 Tesla is a research grade MRI, so all the differentiations that you can see are not used clinically. And so because you can't differentiate between those subnuclei, neurosurgeons rely on stereotactic atlases. So you may have heard of atlas in the context of like a geographic atlas. All an atlas is a map. And so a stereotactic atlas is just a map of neuroanatomy. And so atlases offer the ability and offer the benefit of being in a standardized coordinate system. So this allows you to precisely target the area that you're trying to stimulate. And so the way that atlases are used is by first taking patient imaging. So the bottom picture is an MRI taken preoperatively from a patient. An atlas or map is taken and put onto that patient imaging. And so you use that map in order to segment out regions of the of patient imaging that you can't get from imaging alone. And so the picture on the right are those subnuclei landmarks on a patient image. And so one of the main problems that exists right now in the field is just the sheer number of atlases that are out there. And so these atlases also differ in terms of the delineation of motor and sensory regions. And so some of the reasons for this kind of differences between atlases is that patient demographics, so age of the patient, history, is all contributed to that. Number of patients that they use. Some atlases average across patients, some just use one. Some atlases use MRI and some use histology to make their boundaries. And also some atlases have a different functional or anatomical bias depending on the researcher. And also atlases vary in terms of their detail. So that's kind of the introduction into the field. And so I'm going to then talk about the work that I did comparing thalamic atlases and segmentation techniques. So the research objective that I had was, given the unstandardized delineations of motor and sensory regions across thalamic atlases, the goal of this research was to first quantitatively validate that there were differences between atlases. Next, phase 2 was to determine how those atlas differences affect patient segmentation. And then the last phase 3, was to find which atlases best correspond with clinical outcomes to help clinicians better decide which atlases they should use. So for phase 1, the question was, is there an appreciable difference between atlases? So in order to do this, I obtained 6 atlases from a literature search. And so these atlases differed in their total number of subnuclei, also their total number of motor and sensory subnuclei. And so you could see those 6 atlases pictured to the right of the screen. And then while you can see that there is some sort of similarities between where the motor and where the sensory locations are, they are very different when you start to look deeper. So in order to compare between these atlases, what was first done is comparing the atlases to each other. So each atlas was compared to all the other 5 atlases, including itself, using a registration technique. So just to briefly touch on what registration is. Registration is essentially aligning 2 things, 2 separate images, volumes, objects, what have you, to each other. So in the image to the left, you'll see a 2D registration example. So you'll see a red butterfly wing and a blue butterfly wing. And so the red butterfly wing will be held fixed, while the blue butterfly wing is scaled, shifted, and rotated in order to best overlap that red butterfly wing. And so this was done in 3D to these atlases, firstly. So for example, I took one of the atlases by Alinsky, and I registered that onto the Ewart atlas by scaling, shifting, and rotating that first atlas to best match the second one. And so this was done across all 6 atlases. So what was first done is take all 6 atlases, hold those stationary, and then take another atlas and overlay those onto onto all those other atlases. And so this was done all the way down the line. And so in order to measure overlap, the dice coefficient, or DC, equation was employed. So DC, or dice coefficient, is a measure of overlap. It's twice the volume of intersection between 2 objects divided by the sum of those 2 objects' volume. So if we have 2 things that are perfectly overlapped on each other, the dice coefficient is 1. However, if we start to move those 2 objects further away from each other, the dice coefficient decreases. And then if there's no overlap between those 2, the dice coefficient is 0. So looking at the results of the dice coefficient of this registration, we can first see that there is a line of 1s down the diagonal. And so that makes sense, because if we're registering one thing to itself, it's an identical match, and so we would expect there to be perfect overlap, or a diagonal of 1s. And so when we're looking at this graph, this is showing the overlap of 2 whole thalamus, or whole thalami from 2 different atlases. And so the dice coefficient for the other atlases compared to each other range between 0.7 and 0.9, around there. So while it's not it's not perfect overlap, there's still a really high overlap between those atlases, indicating that across atlases, the thalamus is relatively the same. However, this is not the case when we shift to viewing just the motor regions of the thalamus and compare the motor regions to other motor regions across different atlases. So you can see that the Dice coefficient is starkly lower when comparing motor to motor than when comparing whole thalamus to whole thalamus, and that's the same across all atlases. And so one atlas in particular, the Ewart atlas, showed a significantly lower motor overlap than all the other atlases. And so the same thing was done for sensory thalamus. So the sensory regions were compared to sensory regions of other atlases thalami, and this was done across all 6 atlases. So as you can see, there's still that diagonal of 1s down the middle, meaning that when you're overlapping something to itself, it makes sense. However, when we look at the Dice coefficient comparing other atlases, comparing other different atlases, the Dice coefficient is a lot lower than when comparing the whole entire atlas. And so what this means is that although the different, or although thalamuses or thalami from different atlases show relatively good overlap, the overlap of the motor and sensory regions is much lower, indicating that there's a relative difference between the motor and sensory regions across all atlases. And so to answer our question in phase 1 of whether there's an appreciable difference between atlases, we found that yes, the relative location of motor and sensory subnuclei vary across all atlases. Okay, and so now that we know that atlases are different, our second phase is to determine how atlas choice affects resulting patient segmentations. So when we apply these to patients, what are our results? How are they different? And so in order to do this, a patient's data set of 22 patients with essential tremor was taken, and they all received deep brain stimulation. So this data set was taken from our collaborators at the University of Michigan, and their thalami were extracted from pictures of MRI. Next, 6 atlases, the same 6 atlases that were observed in phase 1 were taken. And these atlases varied in terms of the number of total subnuclei, the number of motor subnuclei, and sensory subnuclei as well. And so what was done is overlapping each atlas onto each patient. So there were 6 atlases and 22 patients. If you do the math, that's 132 patient atlas pairs. And so each atlas was registered onto each patient. And so after the atlas was registered onto the patient, the resulting thalamus was segmented in one of 2 ways. The first way was atlas-based segmentation, or ABS, and the second was DTI-based segmentation, or DTI-BS. And the yellow and green color coding is going to be consistent throughout the slides to help you. And so atlas-based segmentation, or ABS, is the traditional approach when trying to classify the subnuclei of patient thalami. And so in this approach, you essentially use the boundaries of the atlas as a stencil and overlap that onto the patient thalamus, right? So if I was to show you this as an example, so if we had patient data, which is like seen as the points here, and we had atlas subnuclei, which are seen as those colorful ovals, all the atlas-based segmentation method is doing is overlapping one onto the other. And so that's the resulting segmentation that we get. However, the other method that we used was DTI-based segmentation. So just to hit on briefly what DTI is, diffusion tensor imaging, or DTI, is an imaging modality that takes a look at the diffusion of water and uses the diffusion of water in order to determine the tissue properties of the brain. And so this was utilized in the diffusion tensor imaging-based segmentation technique. So in this technique, DTI-BS, each patient had specific DTI imaging that was taken, and then a clustering algorithm was used in order to segment those patients. So in order to show like the same diagram, so if we have those same patient data points, what was done was we used a clustering algorithm that took into account Euclidean distance, which is just like the XYZ distance, and also tissue properties of that patient. And so using these 2 metrics, we then took the atlas, and when the atlas was applied to the patient, it clustered clusters that were closer to each other as being part of the same subnuclei. So compared to the atlas-based segmentation technique, DTI-based segmentation took into account patient features and patient tissue properties when making the segmentation. And so these are the results to the left for just a single patient. And so the Ding 2020 atlas. Ding is a researcher and published an atlas in 2020. And so that atlas was applied to a sample patient. And then the resulting segmentation for atlas-based segmentation, as you can see here, is different than the resulting segmentation for DTI-based segmentation, which you could see there. And so even though it's the same patient, the same atlas, the way that you segment out the patient thalamus is different between these two techniques. And so you can see this was done for all 6 atlases and across all 22 patients. And so in order to compare the differences between these two segmentation techniques, we determined the geometric features of these two resulting segmentations. So the volume and centroid distance was determined for both the motor and sensory regions. And so these are the results just for a single atlas. So on the x-axis, we'll see that this is DNG20, or this is the Ding 2020 atlas. And so as you can see on the right, or on the y-axis, it's comparing motor volume and sensory volume for the left two, and comparing motor centroid distance and sensory centroid distance for the right two. And so each of these graphs shows the differences between, geometric differences between these two techniques, DTI-based segmentation and atlas-based segmentation. And so just throughout, more stars is going to indicate there being a higher difference between these two techniques. So when we look at, and so this was performed across all 6 atlases. And so when we look at the results of comparing these geometric features, we find that atlas-based segmentation generally produces larger motor volume and a smaller sensory volume than DTI-based segmentation. In addition, all sensory metrics were significantly different between the two techniques, and that was across all atlases and for both volume and centroid distance. So sensory was the least reliable between the two techniques. And so each atlas also had at least 3 out of the 4 geometric features be different between the two. And so to answer the question of phase 2, of how does atlas choice affect resulting patient segmentations, the way that we segment out the atlas causes differences to increase. So there are already differences between the atlases. However, when we segment out patient anatomy, these differences increase as the atlases are applied to patients. And so this brings us to the third phase that we explored. And so this, the beginning 2 phases were just determining whether the atlases and the way that they are applied cause different resulting anatomies. However, in the third phase, our focus was looking at the clinical outcomes and seeing how they corresponded with what we observed clinically. So the 2 outcomes that I looked at was tremor reduction, which is the tremor going away, and sustained paresthesia. And sustained paresthesias are basically tingling and that numbness I talked about earlier. And so for a tremor reduction, we would expect there to be a high motor activation and low sensory activation. We're hitting the motor area that we want, and we're steering clear of the sensory area, the sensory subnuclei. However, for sustained paresthesia outcomes, there's a high sensory activation in these areas. And we can't really say anything about the therapeutic effects because although we may be getting a tremor reduction, there's also paresthesia that can result from that. And so the way that the VTA, or volume of tissue activation, was modeled was by first taking an MRI, a preoperative MRI of the patient. In the MRI, the relevant patient anatomy was traced out, and that's the resulting yellow figure you'll see here. And then after that, a CT scan was taken postoperatively, and that allowed the lead to be localized relative to the patient anatomy. After that, diffusion tensor imaging was used again, but in this case it was used for the finite element analysis of the VTA and to make models of electrical stimulation. And so as you can see in the figure at the bottom, here this red around the active contact is the volume of tissue activation. And so in order to determine the amount of VTA overlap of the VTA with the motor regions and with the sensory regions, we developed a metric called the VTA motor overlap percentage. So this is essentially the intersection of the volume of tissue activated and the motor region, all divided by the VTA, and that's times 100. And so this metric is essentially the amount of the VTA that's located within the motor region. And so if we see a high VTA motor overlap percentage, there's going to be a large portion of the VTA that is located in motor region. The same thing for the sensory VTA sensory overlap, but just with sensory subnuclei. And so just to prime you before you see the results, for the therapeutic outcome, we would expect there to be a higher VTA motor overlap than a VTA sensory overlap. So in this case, we would expect the red to be greater than the green, or on the blue on these slides. And so when we look at the results, atlas-based segmentation is on the left. And so as you can see, the red, or the VTA motor overlap, is greater than the blue, which is the VTA sensory overlap for the therapeutic case. And this makes sense, and this is expected for our trend, and this is the same across all 6 atlases. So all 6 atlases show the trend that we are trying to look for. And then if we look to the picture, the graph on the right, for DTI-based segmentation, we see that only 3 out of the 6 atlases show that correct trend that we're looking for of larger motor VTA, or larger motor activation than sensory activation. So in this case, atlas-based segmentation on the left was better than DTI-based segmentation, which is pictured on the right. So this was also undertaken for the side effect outcomes. So because we can't compare the amount of sensory to motor overlap for the paresthesia effect, because as long as you have some sensory overlap, you're going to probably get a paresthesia side effect, we decided to compare the paresthesia outcome to the therapeutic outcome. So we would expect there to be a greater VTA sensory overlap for paresthesia than for tremor. And so in the following graphs, the expected outcome would be that red is greater than green. And so in these graphs, starting at the left, the atlas-based segmentation technique yielded 5 out of the 6 atlases showing the correct trend that we thought, showing that there was higher VTA sensory overlap in the paresthesia case than in the therapeutic case, which was great. However, when we look at DTI-based segmentation on the right, we'll see that 3 out of the 6 atlases show the correct trend that we want. However, 2 out of the 6 atlases show the opposite trend, show that there is higher sensory activation for therapeutic outcomes than for therapeutic outcomes than for paresthesia outcomes, which is not correct. So in this case as well, atlas-based segmentation was much better than DTI-based segmentation. And so just kind of summarizing the results of the clinical analysis, atlas-based segmentation had a higher correspondence to clinical outcomes than DTI-based segmentation did. The Jakob atlas exhibited the correct trend for both clinical outcomes and also for both segmentation techniques, atlas-based and DTI-based. In addition, we observed reverse trends for the VTA sensory overlap when segmented using the DTI-based segmentation method. And so to answer our phase 3, our last question that we had of which atlas should clinicians use, we determined that the Jakob atlas was the best atlas that we observed and aligned best with clinical outcomes for both therapeutic and paresthesia outcomes, segmented both ways, segmented with atlas-based and segmented with DTI-based segmentation. And so some conclusions of all of my analysis, all 3 phases. I found that atlas choice influences segmentation and clinical outcome prediction. Atlas-based segmentation aligned more closely with clinical outcomes than DTI-based segmentation did in this study. The Jakob atlas demonstrated the most consistent clinical accuracy. And so while DTI-based segmentation has potential, it requires further refinement in order to be as effective as atlas-based segmentation is. And so that concludes the analysis portion that I did. And so just to kind of touch on the impact of this work and kind of the translational potential of my findings in the future. So insight from this analysis sheds light on the relative effectiveness of atlases that are currently in use. And so neurosurgeons rely on using these atlases in order to make informed decisions of where they place the lead. So determining the atlases with the highest clinical correspondence is essential because it allows for refined targeting and allows to better target patients, leading to better patient outcomes. And so some of the limitations of the study were that diffusion tensor imaging-based segmentation weighted spatial and tissue properties identically. So maybe changing some of those weights in that clustering algorithm would be better. In addition, exploring other segmentation methods such as neural networks or tractography will be beneficial. So tractography is pictured on the slide to the right. Having a larger patient data set and analyzing more atlases would allow us to make a more informed decision and look at the robustness of our analysis. In addition, combining atlas-based and imaging-based techniques may result in a better segmentation technique. And so I just want to thank Dr. Malaga for all his help and support throughout these 2 years of research. None of this would be possible if it wasn't for his guidance and support, and this work has been so amazing to do, and it's really set me on my path for the future, hopefully being one of the people that does perform these surgeries. I also want to thank the thesis committee for listening to this and also reading through my thesis. I know it was not short. I also want to thank the Computational Neuromodulation Lab for listening to me practice these slides and supporting me throughout this entire process, and to all of you for listening to me present all of this. So thank you all very much. I'm happy to take any questions. Not all at once.
Open the floor up.
Yeah, Maya.
Matthew, can you go back to, in section 1, you talked about how you rotated the images so that they overlap each other to make your correlation factor your DC. Can you explain that process and how you went about doing that?
Yeah, so this was all done computationally. And so it's really just one line of code that does this, but it takes 2 images and then overlaps those images onto each other. It wasn't like spread out through other lines of code. The registration that I used was an iterative point registration. So there's a bunch of registration techniques that exist. And so we looked at linear and nonlinear registration techniques, and then we settled on this one because it gave us the highest dice coefficients while giving us also the transfer function that was used for these registrations. And so it was high dice coefficient, gave us the transfer function we wanted, and that was the reason that we stuck with this one. Yeah, yeah, Keelan.
Great job, by the way.
Thank you.
I was wondering how you chose the atlases that you picked for your study. You said
Yeah.
to them, I'm assuming there were more. I was wondering what criteria you used.
Yeah, so the atlases, so first of all, a literature search was done for which atlases were the most relevant clinically and which atlases were used to make clinically informed decisions. And these atlases kept coming up. And they were also different enough between them. As you can see, like you were here, I go to laser pointer, is different from all the other 2, and that doesn't have this like superior portion of the thalamus being motor thalamus, while the other ones do. So like both a mix of like these are kind of different from each other, and like these are used clinically to do. Yeah. Andy.
I'm interested in the way that should cluster. Like your data, what type of algorithms did you apply? Did you use PCAs?
No, so we used a k-means clustering algorithm, and so this is a messy equation I don't want to put on the slides, but I'll just put it here. So part of it is factoring in like Euclidean distance, and part of it is looking at the tissue properties. So the thing on the right is just a fancy way of saying these are like tissue properties that are similar for like 2 points next to each other. And so using both these, we were able to, this was the distance equation that was used to classify the patient clusters for the diffusion tensor imaging based segmentation technique.
Yeah, and then were there like more than just one? Were there like multiple variables that you have to isolate down to kind of like know like which one is like the biggest contributor?
I set these 2, so this is how like the equation is written, and so alpha is chosen to be default as like 0.5. So and also just this equation weighted like Euclidean distance and tensor dissimilarity, which is tissue properties. It weighted both those 2 equally. So we're thinking of maybe even weighting those 2 sections of the distance function would result in better segmentation outcomes. So, but they were equal for this, yeah. Yeah, Tyler?
So I know in your future work you mentioned you would probably like test more patient samples.
Yeah.
I was wondering like for part A, if you had like access to like patient information, were there like similarities in the demographics, and then like if so, would like different patient demographics correspond to like a better atlas, or you kind of just saw overall that specific atlas worked best for all patients?
That's a great point. It's something I didn't talk about, but we didn't do that like categorical analysis of like patient demographics at all, but like definitely doing so in the future would allow us to do like a better like, if this patient's like over this age, maybe this atlas is a better fit for them. So factoring in that is definitely on the future work, but no, that wasn't done. Yeah, Michael?
So I'm curious, you talk a lot about how using the different atlases, you may have a better idea or be more confused about if you're actually targeting the area you think you're targeting. I know you're kind of alluding to the fact that having a better idea of where you are gives better clinical outcomes. But are there any concerns that if you use the wrong atlas or you're in the wrong place that you're stimulating, that you could see like worsening of the conditions or any other like bad side effects from targeting the wrong area?
So that's kind of like one of the main like problems in this field is that there are so many like different like atlases, and so the clinical analysis that we did, we would expect there to be a higher sensory activation when we do see side effects, but because we're like making those clusterings based on the atlas, those differ and are hinged kind of like on the atlas that we do use to make those like resulting clusters from. So kind of the phase 3 analysis was just like looking at like which atlas should be used. Does that answer your question, or?
Yeah, so if you target the wrong place with
Yeah.
CS, are there issues of like damaging patient health, or is it just
Okay.
going to be less effective?
I see. So the side effects, that's kind of like the remarkable thing about DBS is that you can just turn it off, right? So like if you do target the wrong area, like say you target sensory and you see the paresthesia, they can change actually the place on the lead. I'll actually get an image here, but so the lead has multiple contacts. You can see this has 4 contacts. So if they do this during surgery, so they stimulate the contact at the bottom, and then if that's causing like a paresthesia or tingling outcome, they can move to the contact at the top and basically refine it that way. But yeah, I mean, none of the side effects are permanent because this is reversible. So all the side effects can kind of go away as soon as the implant is turned off. Does that help? Okay. Yeah.
I know, like compared to ABS, IDT wasn't the best method to like search for like the motor and sensory.
Mhm.
But do you think there are other areas of study where IDTBS will be more useful in like searching for certain areas of your brain instead of ABS?
Yeah, so like that's kind of like the hypothesis is that like if we do use like actual patient data in making our clusters, we expect them to be like more kind of specific to the patients and kind of better than just like imposing this like atlas onto it. So there's definitely a lot of potential. There's been previous work that has shown that it's sometimes better at explaining sensory outcomes in the paresthesia case than atlas-based segmentation, but it's so atlas to atlas. It's just different and varies across atlases. So that was kind of the motivation of this work, was to like compare those atlases and see the effects that this has across multiple atlases. So there's definitely room and definitely a lot of work that needs to be done in order to help and use DTI data in order to make informed clustering decisions. But the algorithm that we used didn't show that it had better outcomes. Yeah, Arjun?
So you got that the DTI-BS method, segmentation method, right?
Yeah.
Was that the ABS was better than that one, and then at the end you also mentioned in your limitations that some limitations were that you weighted you weighted the spatial and the tissue
Yeah.
averages, but you weighted them the same.
Mhm.
That was a limitation you put on the DTI-BS. So do you think if you weighted those two things differently, that perhaps you would have gotten different results and maybe the DTI-BS would have been better? Or?
Yeah, so I think that it's definitely like worthwhile to explore that because like this analysis is like changing so many things. So we're like atlases is like an independent variable. Patients are independent variables. So like the fact that there's so many different like independent variables that we're changing in this analysis, we didn't want to add another one of like adding weights to the distance equation. We kind of just wanted to use like one that has been previous done. And this was obtained from previous work done in the field. And there's literature backing this equation. So, but I definitely do think that changing those two properties would like help resulting segmentations for patients. Yeah, it's definitely worthwhile to explore. Yeah, Eddie.
I know you talked about paresthesia as the predominant negative side effect
Mhm.
associated with the sensory DBS as opposed
Yeah.
to motor DBS. Do you still see the other side effects as speech difficulties, even if the electrode is properly placed into the motor region?
Yeah, so that was like kind of like outside the scope of this analysis. So like the other side effects weren't in like the data set that I was analyzing. I don't know like what the relative activations would be for like motor and sensory, but paresthesia was chosen because it was like so obviously like we're hitting sensory when we do get paresthesia outcomes as opposed to like motor, but definitely like that's with further analysis. Yeah, I could definitely answer that question, but I can't now. Yeah.
So this is outside the scope of what you did, but as a biologist, I'm just really fascinated what's known about the underlying cause of essential tremor.
Yeah.
And do they have any idea how the deep brain stimulation is offsetting that?
Yeah, so to answer the first question about essential tremor, it's been implicated to be like both environmental and genetic. And so there's also studies that have shown that like the loss of Purkinje cells in the cerebellum in particular have caused or been associated, correlated with essential tremor. However, for deep brain stimulation, the way that at least researchers are kind of like explaining it is like if you think of like an orchestra kind of like tuning, and every person, like every instrumentalist starts at a different note. When they tune, they kind of tune to the same kind of frequency, and so some sort of like tuning and like pulse of that signal causes the neurons to fire in a certain way that reduces the symptoms. But like the underlying like mechanism is still like unknown. Yeah. Yeah. Yeah.
I'll ask one more question. So you got at the Jacob?
Jakob.
Jacob.
I don't know how to pronounce it.
You got that the Jacob Atlas was kind of the best. And do you think of the atlases that you considered, do you know anything about how Jacob made his atlas that favored it and for you to consider it to be the best? Like, was there any particular method Jacob did to make his atlas that you think might have been good and better than the other?
Yeah, it's an interesting question because like me and Dr. M have like spent a lot of time like trying to like figure out like why is it that like certain atlases like perform better than others, because Jacob doesn't have the most number of subnuclei, it doesn't have the least, it doesn't have the most motor, it doesn't have the least sensory, or the least motor, most sensory, least sensory. So one of the potential reasons is that it's maybe in that sweet spot. But yeah, I don't know. I could tell you that it was made from histology. So the Jacob Atlas is kind of an offshoot of another atlas made from histology. And so another thing about Jacob's atlas is that the relative regions of the subnuclei are relatively kind of like globular, is like another component of this. Like some have like very thin regions and like that are stretched out. This you can see like they're kind of like all generally a similar like shape and similar volume. So that maybe will have something to do with it. But yeah, aside from that, I don't know specifically why. Yeah, Dr. B.
Really great talk, Max. Congratulations.
Thank you.
And hopefully you can sleep well tonight. I don't have a technical. I do have a lot of technical questions. I'll ask Dr. M later if he's thought about using ultrasound. Anyway. Would you? Yeah. Would you consider, like, would you undergo DBS yourself in the state of the art?
Oh, I 100% would. It's interesting because like the same technology is being used in spinal cord stimulator implants for like pain management, and I've been trying to convince my grandma to get it, and she just doesn't want to get it. But like if I had like really severe pain or like a movement disorder, I would 100% do this because one, it's reversible. You can easily turn it off, and it's not as if you have to like remember to like take medication. And so yeah, this is just, and they keep putting out more and more like advances, and like, so they made MRI friendly leads, and like they keep advancing it, and it's getting better and better. So yeah, I would definitely get it. Yeah.
Should have got this many questions in class. Yeah. I think we're gonna call it. So thank you very much, everyone, for coming out.
Oh,
One more.
Oh, one more. Oh,
Sorry.
dang. Sorry.
Yeah,
Go ahead.
no, sorry. Okay, sorry. Kind of building off Arjun's question.
Yeah.
So, if you even like looked into this at all, why do you think that the diffusion tensor imaging had like a lower correspondence to the clinical outcomes in ABS? Like, I don't know. We do a lot with DTI and linguistics, but I don't really know I don't really know the aspects of it in like what you're doing in your work. So.
Yeah. I think the main thing is kind of the refinement of the algorithm that we're using. So that's kind of a whole project in and of itself, is just like parsing out the correct kind of like, are we valuing like tissue properties too much in like in this region, or should we value mostly like the kind of like the distance metrics more in like other parts of like the thalamus, or like different atlases? Should we like change the weights of those two different parts of like the algorithm? So that's definitely like an ongoing kind of like question, and I really don't know why it's, yeah.
All right, now for real this time. Yeah. Let's